Well, hello. So we are back to journey our way through bacteria. Okay, so we looked at viruses the other time. We're now going to look at bacteria. Okay, so with bacteria, they belong to the kingdom Monera. So either you say kingdom prokaryote or kingdom Monera. It contains the oldest and smallest of living cells. In fact, the smallest bacteria currently is the mycoplasma. It's known as mycoplasma, right? It's known to be smaller than the smallest virus. So regarded as the smallest living organism on Earth. Mycoplasma is the smallest bacterium, as I've said, without a cell wall. Many are quite harmless, but few can be infectious, affecting the respiratory, urinary, and then genital tracts. Some 200 types have been identified. As I said, mostly are not that contagious, but when they decide to be, or those that are contagious, it can be really, really deadly and leads to a lot, especially in respiratory situations, coughing and sneezing quite a lot. All right, so that's just a little bit about the mycoplasma, the smallest living organism, which happens to be a bacteria. Okay, I don't know whether we should be clapping. Well, let's move on. Usually they are single-celled. They have no true nucleus or membrane-bound organelles. Okay, the nucleus, we usually say that it's no true nucleus because it doesn't have membranes surrounding it. Okay, they are quite ubiquitous. They are everywhere. They are involved in a lot of processes. In fact, they are equally as harmful as they are, helpful. Okay, right. As they are helpful, they are involved in a lot of industrial and ecological and then medical issues. Okay, so they are quite harmful and then they are quite useful as well in terms of their economic importance. So bacterial diversity and classification, as you have said, that's the Monera. They have some subkingdoms, Archaebacteria. They have unusual structures and habitats. They have the eubacteria, which is the true bacteria that we usually refer to when we are talking about bacteria. Okay, so if archaea, what's their habitat? Some are in methanogens, some are in extreme halophiles, and some are in thermoacidophiles. So methanogens usually they capture hydrogen and CO2 and convert it to methane. And they live only in anaerobic environments. In those in extreme halophiles, they are salt-loving. They live in areas of high salt content, deep sea, and then great salt lake. So such conditions, we usually don't find organisms because the hypertonicity of the environment, which can actually kill these organisms. The thermoacidophiles, they are in acidic and extreme temperature environments, very, very high temperature environments. We do find these bacteria, and they are Archaebacteria. So in volcano vents, we are likely to find them there. So with the eubacteria, we I want to look at some of their characteristics. So you can distinguish them in two ways with reference to their cell walls. You can use a test called a gram test. Okay, a gram test to actually determine or distinguish between them. So you can have the gram positive bacteria and the gram negative bacteria, and this is important in medicine or medical diagnosis, so that you can distinguish between and actually pinpoint exactly which bacteria is causing harm to someone. Okay, so with the gram positive or with the gram staining or in gram positive, you have the cell retaining the stain and appears purple when it's positive, uses crystal violet, and then gram negative, they are not able to then are not able to retain the stain, and when saffron is added, then it gives a pink or a color. So let's take a look at how the staining is done. So for gram staining, what first happens is that there's a smear, bacterial smear, placed on the microscope slide, and then crystal violet stain is added to the smear, right? That is followed by iodine treatment, where iodine solution is also added to the slide or to the smear on the slide. Okay. And this iodine and crystal violet stain forms a complex, so that it becomes sort of the idea is to sort of retain the stain within the cell wall or within the bacteria, specifically the cell wall. Okay. Then later, the complex is treated by adding alcohol or acetone. Okay, alcohol or acetone. This is supposed to wash off the stain from the bacteria, right? And what again happens is that because the gram positive, the cell walls are thicker, and having a material, an acid called teichoic acid, and the network of the peptidoglycan, that's the materials that form the cell wall, is so complex. It's able to then retain the iodine crystal violet stain, giving it a purple color. Now, to attest whether the stain, the crystal violet and iodine complex, that stain has been well retained within the cell wall, after washing off with the acetone or the alcohol, saffronin is added to the complex or to the smear on the slide. And if it's a gram positive bacteria, the saffronin will not have any effect because the crystal violet and then the iodine stain has been locked in within the cell wall, so there will be nowhere for the saffronin to stay within the cell wall, so it doesn't stain the cell wall. However, in gram negative, because the iodine and crystal violet stain has been washed off because the cell wall is not thick enough and all that, the saffronin that is added later then stains the cell wall, right? So that would give a differential in terms of the color of the bacteria. So the gram positive bacteria, as you can see here, is having the purple or violet color, whilst the gram negative, due to the saffronin that was added later, turns it pink, right? Gives it a pink color. It stains it and then becomes negative, pink or red color. So that's one way you can differentiate between different types of bacteria, and it's high grade when it comes to diagnosing diseases in terms of if the causative agent is bacteria. Then we go to autotrophic or heterotrophic. Some have been identified to be producing their own food, while others obtain food from other sources. Okay, let's move ahead. So this is the process: adding crystal violet, adding the iodine solution, washing with alcohol, application of the saffron. We move on. This is a diagram that is showing you the various layers of the bacterial wall. So there is the cell wall. The material that forms it mainly is the peptidoglycan. Okay, so that is it. So this gram negative, this gram positive. So gram positive, you see that there's the wall is thicker, and if you compare the peptidoglycan layer, see it right here, it's very small in the gram negative, but it's very thick in the gram positive. Okay, and then the gram negative, outside the cell wall, it has the outer membrane. All right, okay, and so on. So that's of interest, that's of interest so far as the gram staining is concerned. Okay, so you also can you have the classification by their using their shapes. So you have the rod shape, which is the bacilli. Okay, bacilli, that's the plural. One is bacillus. Then if it's spherical shape, you have the cocci or coccus for one. Then you have other shapes like the spiral shaped or the spirilla. Some also call the spiral sheets, like the corkscrew used to remove the cork from a wine champagne bottle. We also have other terms, still looking at their structure, as diplo. Diplo is a prefix that refers to a paired arrangement of cell growth. So for example, if you have two spherically shaped bacteria, then you say that diplococci or diplococcus, right? To mean that we have two spherically shaped bacteria together, right? So that's diplo. All right. You can also use it for where you have the rod shape, right? You can say diplobacillus. Okay, when you have two rod-shaped bacteria together. Okay. So usually where you have those accumulation, two or more bacteria, spherical or rod-shaped, we mainly have the rod and then the spherical. You mainly have the rod and then the spherical. Okay, then we also have staphylo. So you can say staphylococcus, staphylobacillus. Right? It describes an arrangement of cells that resemble. Okay, so it's a group. Usually it's a group, right? They clump together. Then we say staphylo. So you can say staphylococci or staphylococcus. Okay. So this is just two. And this is a cluster or so arrangement of cells. Then you can also have strepto. Strepto is a prefix that refers to an arrangement of chains of cells. So you have a long chain, maybe 10 chains of spherically shaped bacteria. Then you say streptococci. If they are rod shaped, you can say strepto what? Those streptobacillus or bacilli or bacilli. Okay, so that's how we name them. So just a little bit about the gram positive and negative again. So you see, this is the negative. You see, so it's red or pink. It's not purple. Then gram positive is purple or violet due to the violet stain that was added together with the iodine that was retained within the cell. Then, how do we identify this one? Or is it gram positive or gram negative? What do you think? Yeah, so if you say gram negative, you are there. bacillus. Okay, so you can see that it's rod-shaped, right? Rod-shaped, and they are in the form of chains, aren't they? They are in the form of chains. So that will be streptobacillus, streptobacilli, strepto. That's in the chains of rod-shaped bacteria. All these be positive, negative. It's positive, isn't it? Gram-positive, because they are violet. What will this be? So gram-positive, definitely. You can see the purple column. Okay, so let's look at other classifications of bacteria or groups of bacteria. They are the cyanobacteria, photosynthetic, using chemicals to capture sunlight, once known as blue-green algae, low nucleus or chloroplast. Too many of certain types cause eutrophication, which are population blooms. This causes oxygen to be used up in bodies of water. There are so many of these in water. They tend to sort of make the turbidity of the water so low, and then or so high, and then it's difficult for even light to penetrate. You only have life at the top of the water and so on. There are so many algae in the water, and that's what we mean by eutrophication. So that's an example of a cyanobacteria having the chlorophyll characteristically showing due to the presence of chloroplast. Okay, so they are photosynthetic. They have spirochetes, which are also they are similar to the spirilla, but they have a more rigid spiral shape, if you like. So gram-negative, spiral shape, heterotrophic, aerobic or anaerobic, move with a corkscrew motion, live either freely, symbiotically, or parasitically. Example: Treponema pallidum causes syphilis. So I have more examples of gram-positive or characterization. All but a few of these things positive. Example includes streptococci, which causes strep throat, and lactobacilli, which helps make yogurts, right? So this is the helpful one that we talk about. Actinomycetes are gram-positive form that lives in soil and help produce and for production of antibiotics. You have the true form, that's the proteobacteria, which are the largest. They are in 3 divisions. You can have the enteric bacteria, the chemoautotrophs. Then we have the nitrogen fixing bacteria. So enteric means either relating to or inside intestines. So we have some gram-negative. Of course, they are heterotrophic, aerobic or anaerobic. Example is Escherichia coli, E. coli, Salmonella. Okay, Escherichia coli, E. coli, and then Salmonella. Okay, then we have for the chemoautotrophs, gram-negative. They can extract energy from minerals and use to produce food, so they get that energy, just like chlorophyllous organisms absorb energy from sunlight, and they use it for photosynthesis. Chemoautotrophs also do the same, but they get the energy from chemicals to make food, right? Nitrogen-fixing bacteria, they are also gram-negative, living freely or symbiotically. Example is the rhizobium. All right, so we now get into the structure of the bacteria. So typically, bacteria have a cell wall. It may have a cell wall or not. Majority have cell wall. They have cell membrane, which characterizes is due to no mitochondrium, cytoplasm. No membrane-bound organelles are found within the cytoplasm. So some have outer cavities called capsules, pili, or fimbria, that adhere them to the host. Good. So this is a diagram of two different diagrams of or similar diagrams of the bacteria, showing you. So you have the ribosome. The ribosomes are smaller than that in the eukaryotic cells, plant, animal cell, fungi, and so on. You have nucleoid, secular nucleoid or chromosome, DNA. Okay, there's a pili which is connected to the cytoplasm. You have the cell membrane, right? Those structures, that are connected, that are not connected to the cytoplasm, like hair-like structures found like on the cell wall. That's the fimbria, right? One is fimbrium. Fimbrium. So, and then we have the cell membrane, of course. The plasmid is a smaller DNA, flagella, can be one, or as you can see, 2 or 3 or more in some organisms. You have a cell wall, a cytoplasm. So basically, that is that the structure that this one is not having is the mesosome, which is known to be some kind of an invagination of the cell membrane, right? Which some have said to be involved in, energy production, like functioning like the mitochondria in eukaryotic cell. But it's not widespread. It's not very popular. So that's it. So some will have some slime capsule and so on. Okay, then we have, of course, the flagellum is one here. So basically that's it in terms of the structure. Okay, so we go to looking at this typical bacteria. So the Escherichia coli. All right, you have the Escherichia coli. Having the basic features that we expect the bacteria to have. Okay, the basic features that we expect the bacteria to have, all present. So as you know, the cell wall gives structure to the cell, and also protects the cell. Protect the cell. Most of the bacteria, especially the enteric ones, the parasitic ones, they will definitely come against a lot of obstacles from the host, which will try to prevent itself from being attacked by the bacteria. So they are there. They are structured to prevent the bacteria from being sort of attacked and destroyed. So most antibiotics are designed, therefore, to actually counteract the cell wall of the bacteria. When the cell wall is so tough, so strong, is permeated, definitely then it means that the bacteria is exposed, right? It's exposed, and then can be attacked by the host white blood cells. It can be infiltrated with water. Water can fill the cell, which can cause it to burst and all that. So the cell wall is very, very important when it comes in terms of medicine and in the health sector. Okay, then we can also talk about there's some slime layers that we earlier on mentioned that also offers protection, attachments, and nutrients, serve as nutrient reserves. Okay, the usual functions of the cell membrane, we can go back a little so that we can talk about the general features and their functions. The cell membrane retains the cytoplasm or protoplasm, selectively permeable, as you know, allowing transport of certain materials in and out. Contains receptor proteins for environmental signaling, so on. Then we mentioned the plasmid, right? The plasmid, which is a strand of DNA circular. Okay, that transfer DNA from one bacterium to another, like during reproduction, involving drug resistance mechanisms, produce virulence factors, produce bacteriocins that can kill other bacteria. Then we talk about the nucleoid, right, which is the main DNA circular donor. So it's usually haploid, normally one copy per cell, looped and coiled. Nucleoid means aggregated DNA. Its function is to contain genetic information or genes encoded to make proteins or to develop parts of the bacteria. Then we talk about the pili, as we have generally said, for attachment to surfaces, to the host and so on, cells. And very important when it comes to reproduction, right? Reproduction of the bacteria. It attaches to another bacterium in the conjugation process, that is sexual reproduction, though they hardly do that, but it happens, in some bacteria where they undergo conjugation. So the pili attaches the one bacteria to the next, and there can be exchange of genetic materials from the cytoplasm through the pili to the next bacterium. Okay, right. Then the fimbria, as I've said, this small unit here, like hair-like, hair-like. Let me just use a pen to just circle that area. Yeah, so this one here is hair-like protein in nature, and attaches it to the to surface, shorter than the pili. Usually they are fine, numerous hair-like appendages. Okay, as I've said, the flagellum, that is involved in mainly locomotion. The flagellum or flagella involved in locomotion, movements, made up of, so it's a very complex process made up of protein, some part of the protein, and long parts, the filaments called flagellum. It has a hook and it has a extended body, which is within the cytoplasm and the cell wall of the bacterium. Depending on the number, you can have different names for it. If it is one, we refer to it as monotrichous. There are two. If it's one on both ends, so if you find another flagellum on this side, okay, and then another one, or for this one, for example, there's one here, then you add another one there, then it's polar, right? One on opposite ends, and that's polar. So that's how it is called, right? Where it is two, then you say amphitrichous, like amphibian. Then where there are two, we have lopho. Where we have a cluster, so many at one end, then you say lophotrichous, lopho, Lopho, right? And where they are all over the surface of the bacterium, then we say we have the petrichous, like is that trying to say periphery, you know, like on the surface. So many of them, they have like that. Okay, so basically that's the structure. So why don't we move on, right? So we talk about all these things, the flagellum, and then how it moves the bacteria. We have also said that some are photosynthetic, you know, microbes live in the guts, those others that are parasitic and all that. Alright, so we use other classifications or terms for bacteria. Say that some are saprophytes, they feed on dead or decaying matter. They have this is supposed to be autotroph, okay, so that we have autotroph uses sunlight, chemotroph use chemicals. Then we have obligate anaerobes, they cannot survive in oxygen. Then we have obligate aerobes, cannot survive without oxygen. We have facultative anaerobes, can survive with or without oxygen. So take note of some of these definitions or terms. Thermophilic, bacteria that grow best between 40 degrees and 110 degrees. Wow, that's extremely high. But they do well, as you have said, in the volcanic conditions and so on. Most bacteria prefer acidic environment of 6.0 or lower. Okay. Bacterial reproduction. So bacterial reproduce asexually by a process known as binary fission, right? Binary fission. So think for now the video is long enough, so I want to pause here so that we can digest on the structures, so basically the structures and the functions of the bacterium. Look at the different the structures that they have, and then relate them to their functions. We talk about the spherical shaped. We talk about as a coccus. We talk about bacillus as a rod shape. We talk about the spiral sheets, shaped. There's even a comma shaped, like the bacteria of the cholera, right? So we shall have a separate video on reproduction to talk about binary fission, and then some other types of sexual reproduction and reproduction that's in the bacteria. So I'll see you in the next one.